And thank you, Dennis, for that kind introduction. And yes, I've been very lucky that I have very, very good mentors in my life. And hopefully I will get to repay that favor. So I'm going to tell you a little bit about marine natural products as potential treatments for pancreatic cancer. And at first, we're going to start with telling you what a natural product is. And these are compounds that get made by organisms which are not essential for the organism to survive. Because they take energy to be made, we assume that they confer some sort of advantage to the organism by making it. So that's the reason they make it. They also are called secondary metabolites. So whenever you hear natural product or secondary metabolite, that's what we're talking about. So why explore the oceans for medicines? A lot of the drugs, over 62% of the drugs that we currently use, can be traced back to a natural product. Some examples are aspirin, morphine, penicillin, and Taxol. And Taxol is the most used drug against cancer right now. And of course, all of these are of terrestrial origin, but the oceans cover over 70% of the Earth's surface. So it makes sense that we found this many from terrestrial sources. We're going to find more from the ocean. There are more than 200,000 described species of plants and animals in the sea, and there are many left to be discovered. Of this, the small percentage that have been studied have already yielded 10,000 novel chemicals, but only a fraction of this has been tested for pharmaceutical potential. So as you can see, this is an untapped source of potential new drugs. Some examples of drugs that came from the sea that are used against cancer are Yondelis, or actinicidin 743. This is an anti-tumor drug, and this is a structure, and this is what the C-squared that it comes from looks like. It's an anti-tumor drug from actinicidin turbinata, or a C-squared or tunicate, that is found in the Caribbean and Mediterranean seas. It's also found off the coast of Fort Pierce. You can get it right here. And actually, Dr. Amy Wright helped find the structure of this compound. And that's very important because without that, you cannot get the synthesis and make it in the lab. So it is very essential that we always know the structure of the compounds. And it has been approved by the European Commission for treatment of soft tissue sarcoma and ovarian cancer. And it is pending FDA approval for the treatment of soft tissue sarcoma. Another example is halaben or eribulin, which is a fully synthetic analog of the marine sponge natural product helicondrin B. Eribulin prevents cell division, leading to cancer cell death. And it was approved by the FDA in 2010 to treat patients with metastatic breast cancer that had already received 2 rounds of chemotherapy before that. In the European Commission, they actually approved it based on the results that they had seen to treat patients that have received only 1 round of treatment, and they're actually looking into making it the first type of response that they will use for breast cancer because they have seen a lot of very good results with this drug. So I'm part of the marine biomedical and biotechnology research here at Harvard Branch, and our mission is to harness the power of marine biotechnology to improve the human condition through the discovery of better drugs, alternative fuels, and industrial chemicals. And we're composed of 3 groups. We have a natural products chemistry group that is led by Dr. Amy Wright. And this group does the isolation and purification of secondary metabolites. They do the chemical characterization of those compounds, so they figure out the structures. And they have worldwide collaboration to test against dreaded diseases, from infectious disease to many different kinds of cancer. We also have a marine microorganisms group that is led by Dr. Peter McCarthy. And this does the isolation and culture of marine microorganisms. And they also do biotechnology applications. They're trying to find enzymes to produce alternative fuels and things like that. My group is the cancer cell biology group. And so what we do is we screen these compounds for bioactivity. And once we have an activity, we try to figure out how the compound does its activity or the mechanism of action. So you will hear that phrase quite a bit. And because at some point I'm going to toss these words at you, I wanted to introduce these concepts so that you know what I'm talking about. So 2 key concepts in cell biology are apoptosis and signal transduction. And apoptosis stands for programmed cell death. It is a normal process in which a cell that has become infected or has finished its function or is an older cell can kill itself in a very controlled way. This is, as I said, it's a very normal process. But in cancer, this process is disrupted, and it allows cells that should be dying to survive. Most cancer cells are resistant to apoptosis. So one easy way to think of apoptosis is we all have experienced infection. And normally when you have an infection, one of the ways the doctors know about this is because your white blood cells counts are elevated. And that's because your white blood cells are responding to that infectious agent and they're trying to destroy it. But once they've done that, they kill themselves to eliminate the excess of T cells or white cells to just go back to normal because you don't need them anymore. So that's one easy way to think of apoptosis. So now I'm going to move on to signal transduction. And signal transduction, Wikipedia says in biology, is simply the process by which one cell changes one kind of a stimulus into something else. And cells kind of use signaling cascades. They activate something, that activates something else, that activates something else to do their function. So that's how they divide. That's how they produce factors that allow them to grow or to produce new blood vessels. That's how they undergo apoptosis, through these signaling cascades. And an easy way, Amy came up with the best way to think about signal transduction. So if you call to order for a pizza, the delivery guy gets on his car and he follows all the signaling and everything works out, you got your pizza. Everybody happy. However, if he gets on his car and he tries to follow the signals, but he gets to a signal that is not working, what's going to happen is that he gets into an accident, no pizza, this is bad. So if we put that in terms of a cell now, a normal cell, all the signaling pathways work. So we get pizza. So only proliferates when it's needed, it can undergo programmed cell death, and it only grows in its tissue of origin. A cancer cell has defective signaling pathways, so those lights don't work. There's proliferation signals that are always on. The cells cannot undergo apoptosis, and this cell is able to grow on other tissues. And that's a characteristic of cancer, because a skin cell should never like to be growing in your lung. So cancer cells are very abnormal that way. So I've been telling you about cancer, but I always like to make this point that there's not a single disease that is cancer. Cancer is a collective name that we give to a group of more than 100 diseases. What they have in common is that you have cells that are growing uncontrollably, and that these cells contain damaged DNA. The way they acquire that damaged DNA could be through inheritance, could be exposure to chemicals, it could be infections, it could be smoking, it could be excessive sun exposure. So there are many factors that could cause that. And we believe in the two-hit theory. There's two events that normally lead to the cancer to start. My group focuses on pancreatic cancer, and the reason we focus on this disease is that it is the fourth leading cause of cancer death in the US. Patients that are diagnosed, only 6% survive 5 years past that diagnosis. So the drugs that we're using currently are not very effective. And we have made progress with many cancers in which we have reduced the incidence and the lethality of many cancers, including breast and prostate cancer. But the incidence and lethality rates for pancreatic cancer have been increasing in the last decade. So we really need new treatments to treat this disease. So some characteristics of pancreatic cancer. We know that there are no early detection methods currently exist. We don't have an easy way to diagnose it. The tumors are highly metastatic. That means they spread to other organs. The tumors are resistant to apoptosis. And because most chemotherapies work by inducing apoptosis, the tumors are resistant to chemotherapy. We know that pancreatitis, or inflammation of the pancreas, seems to increase the risk of developing pancreatic cancer. We know that pancreatic cancer cells are able to activate very unusual pathways to provide nutrients to the growing tumor. And we know that pancreatic cancer uses the microenvironment surrounding it to grow. So this leads us to put together a model of pancreatic cancer progression. So if you have a pancreatic cell and you expose this cell to chronic inflammation, what's going to happen is you're going to activate signaling pathways such as STAT3 and NFKB that promote survival and proliferation. And you're going to cause this cell to release soluble factors such as IL-8 and CCL2. These factors attract cells of the immune system, And these cells of the immune system make it to the pancreas, where they themselves release soluble factors. They release angiogenic factors, factors that promote the growth of new blood vessels, and they release growth factors, creating a microenvironment that is very rich and that facilitates cells that are abnormal to grow. Chronic inflammation also causes cellular turnover, and that increases the chances of a mutation occurring. So if you have a mutation occurring in this microenvironment, you're going to lead to a cancer cell, and once you get one cancer cell, this microenvironment is going to allow it to grow and form a tumor. Now I'm going to put it in a scary form, so please bear with me, and I will explain this. We like to think these terms. So I told you inflammation causes the release of soluble factors. Now I added a few more. But what we know they do is they activate signal transduction pathways such as NFKB, STAT3, RAS, and RAGE. These pathways help that inflammation to persist, and they also activate cellular events such as proliferation, resistance to apoptosis, activate unusual pathways to get nutrients such as macropinocytosis, autophagy, and the growth of new blood vessels such as angiogenesis. They also activate pancreatic stellate cells. These are normal cells that are around the tumor, but they release soluble factors that create that microenvironment. They attract cells of the immune system that, again, help to perpetuate that inflammation, and they help to create that microenvironment. And so when you have all these events occurring, it leads to tumor initiation and then to metastasis. So this is scary, but for us, this provides targets that we can go and say, can we act at these different levels and find inhibitors for this disease? So we were very lucky that for many years we had access to a ship and a sub that allowed us to collect marine invertebrates. And from these, our chemists made extracts. So if you have coffee or tea, you're making an extract. Our chemists use something more harsh than water to make these extracts, but it's the similar process. And then they fractionate these extracts, and you can think of every single peak that you look at here as a compound to create what we call the peak library. And these are enriched fractions that contain a mixture of 2 or 3 compounds. They send this to my lab where we do screening, and once we find a bioactivity, we send it back to the chemists who isolate the pure compound based on the bioactivity, and then they send it back to us so we figure out how it works or do mechanism of action studies. So we do what is called phenotypic-based screening. We're testing the ability of compounds to change a signaling pathway. If something is always turned on in cancer, we try to see if we can turn it off, and vice versa. If it's not turning on, can we restore that? All our assays are cell-based, and we follow what we call a forward chemical genetics approach. We know we're changing a phenotype. We don't know where in the pathway we're causing that change, so we have to do mechanism of action studies to figure that out. So this is a busy slide, but I just want to make a point that we have 3 main areas that we focus in my lab. We look at molecules that regulate cell proliferation and survival, and these are some of the molecules that we look at. We have molecules that regulate inflammation, and we have molecules that affect mast cell degranulation and migration. And I'm going to tell you a little bit about the progress that we've made in these 3 areas. So first, we're going to start with the marine natural compounds that target molecules that affect or that regulate inflammation. Inflammation is a very normal response. It's how your body tells your immune system that you have gotten an infection or that you have gotten an injury. So you want that to be there. It's only when it becomes what we call chronic or constant that becomes a problem, when it is not resolved. Because when it becomes chronic, then you get tissue injury through the swelling. You get a microenvironment that facilitates tumor growth. And we have constant damage and repair, which increase cellular turnover, which increases the probability of getting a malignancy. Most adult cancers are frequently preceded by inflammation. Long-term users of NSAIDs, such as aspirin, have a 40 to 50% reduced risk of developing colon, lung, and stomach cancers. And we know that patients with pancreatitis have an increased risk of developing pancreatic cancer. Those that have hereditary pancreatitis have a 53 times higher risk of developing pancreatic cancer than the average population. And those that have sporadic pancreatitis have about a 17 times higher risk than the normal population. So I told you that inflammation caused the release of all of these soluble factors and activation of all these signaling pathways. But for us, those represent targets that we can go after. So the first thing we want to see is, can we find inhibitors of IL-8? And so the way we did this is we have pancreatic cancer cells in the lab, the BXPC3, that produce high levels of IL-8 without us doing anything to them. So I plate those cells and allow them to adhere overnight. The next day, I replace the media with fresh media and the presence or absence of marine compounds. I incubate these cells for about 5 hours to allow for new IL-8 production. And then I take the media where the IL-8 has been released, and I can measure that through an ELISA. And then I lyse the cells and mix it with ethidium bromide, and that gives us an idea of the DNA content in those cells, and we use that as a measurement of cell viability. And an ELISA is an enzyme-linked immunosorbent assay. And the way it works is that we coat a plate with a capture antibody. This traps the cytokine or soluble factor. And then we use a different antibody, which is called a detection antibody, that also recognizes this soluble factor to kind of create a sandwich. And then once we have that, we can use horseradish peroxidase and a mixture of substrate that produces a color change, and we can follow that on a plate reader. And so that's a way to quantitate how much of that soluble factor or cytokine was made. And through that effort, we found thiopederine K. So this is its structure, it's quite complicated, and it comes from the marine sponge Discodermia. It's a known compound, so we knew that it had cytotoxic activity against tumor cells, but the inhibition of interleukin-8 is a new activity for this compound. And the IC50, or the dose to see 50% inhibition for interleukin-8, or for thiopederine K, is 370 nanomolar. The IC50 for IL-8 inhibition for curcumin is 28 micromolar. So curcumin is a compound that is contained within the within the spice turmeric, and it is a known anti-inflammatory compound, but you can see how much more active this marine compound is than curcumin. So we're pretty excited about that. The reason we're excited is because interleukin-8 promotes the growth of new blood vessels, and it also facilitates metastasis. So the inhibitors of IL-8 may be anti-metastatic and anti-inflammatory. We also have an assay to screen for inhibitors of NFKB, which is an important factor in regulation of inflammation and apoptosis. And the way we do this is we have an engineered lung cancer cell line that contains a luciferase reporter gene for NFKB activity. So we give TNF-alpha to the cells, which is going to cause activation of the NFKB pathway, and NFKB is a transcription factor, so that just means it makes it produce genes. And so it will produce all its normal genes, but it will also produce luciferin. And when we combine that with luciferase, it produces light that we can follow through a plate reader. And if we have an inhibitor, there will be a decrease on that light, and we can find it. And through this assay, we found SPONGIATRIOL. SPONGIATRIOL has this structure. It comes from the sponge. It has known anti-tumor and antiviral activities, but inhibition of NFKB is a novel activity for this. And these here are cancer cells. This is flow cytometry results. But if you look at this line, anything to the right of that line are cells that have active NFKB. And these are pancreatic cancer cells that have active NFKB. But if I treat them with 2 times the IC50 for SPONGIATRIOL, you can see that I have a lot less of the active NFKB. Now, NFKB is important because it not only regulates inflammation, but it also it blocks apoptosis from occurring. So we knew that pancreatic cancer cells have constitutive levels of active NFKB in them. And so if we were blocking NFKB, we should be able to induce apoptosis in these highly resistant cells. And indeed, SPONGIATRIOL is able to induce apoptosis in 2 types of pancreatic cancer cells. So that's very exciting results. Through this type of assay, we also found microsclerodermin A. Microsclerodermin A has a much more complicated structure, and it comes from a deep water sponge of the genus Microscleroderma. It has anti-tumor, antifungal activity, but again, Inhibition of NFKB is a total novel activity for this compound. It has an IC50 that is 1.2 micromolar, and it induces apoptosis in ASPC1s, BXPC3s, and PAN1 cells. So this one is also able to induce apoptosis in all our resistant cell lines. But I keep on talking to you about apoptosis, and I think I should show you what it looks like. So we were very lucky that our management about 6 months ago got us a high-content imager that allows us to take images very fast of our cells and also quantitate different cellular events. And through generous donations, I had the means of buying fluorescent reagents that allows us to capture this event. So these are pancreatic cancer cells. They have been labeled their nuclei in blue. If they are undergoing apoptosis, they are going to cleave a molecule called caspase-3, and it's going to look green. And if they die, their membrane will lose its integrity, and they're going to be able to absorb a red dye. So apoptotic cells are going to look blue, then green, then red. Oh, thank you. And so we took pictures every 20 minutes for 16 hours. And you can see that there's a few cells undergoing apoptosis. This is our methanol vehicle control. Now I'm going to show you what microsclerodermin A does in these cells. So again, remember, apoptotic cells, blue, then green, then red. Sorry, that's wrong. So we're very excited about that. You can see how much active our compound is. And I wanted to use that as my Christmas card, but people thought it was too geeky. But for inhibitors of inflammation, we have so far, for NFKB, we have found 9 pure compounds that inhibit these pathways, and 70 fractions corresponding to 23 different organisms that require further purification. For STAT3, we have found 20 fractions corresponding to 15 different organisms that require further purification. For interleukin-8, we have found 10 pure compounds that are able to inhibit IL-8. We're currently screening for RAGE, which is a receptor that is involved in regulation of inflammation. And the screening is ongoing, but so far MANZAMINE A is kind of showing activity, and I'm going to tell you a lot about MANZAMINE A, and hopefully you will be as excited about this compound as I am. So our conclusions for inhibitors of inflammation in pancreatic cancer: inflammation plays an important role in both the initiation and progression of pancreatic cancer. And we have found marine natural compounds that are able to inhibit important inflammatory signal transduction pathways. These compounds have the potential to not only treat pancreatic cancer, but also to prevent it from ever forming if it's given to patients with pancreatitis. So we're very excited about these compounds, and we're hoping to submit a larger grant to screen for more active compounds and also to further our understanding of the compounds that we already have found. So now I'm going to tell you about marine natural compounds that target mast cell degranulation and migration. And mast cells are very important in pancreatic cancer. So when you have a pancreatic tumor, it's normally surrounded by what we call the stroma, which is made out of connective tissue and healthy neighboring cells, but they contribute to that microenvironment that kind of facilitates the tumor growth. We know that the tumor cells release CCL2, and that attracts mast cells to migrate to the tumor stroma. Once the mast cells get there, they degranulate, and they release molecules that cause blood vessel formation and that favor proliferation, and these in turn help the tumor to grow and metastasize. Again, when I present these pathways, we see opportunities to target things, and so we thought about finding compounds that block the release of CCL2 to prevent the mast cells from ever migrating to the pancreas, or finding compounds that block degranulation to prevent the release of those factors. And those compounds will also have utilities for other diseases, because mast cells, for those of you that suffer allergies, they are the ones responsible. They're the ones that release the histamine that gives you your runny nose and your watery eyes. So the first thing that we did was create a CCL2 ELISA to try to find inhibitors of this. We use a different kind of pancreatic cancer cell line because this one produces large levels of CCL2 just by themselves. We don't have to do anything to them. So we plate the cells, we allow them to adhere overnight. The next day we replace the media with fresh media in the presence or absence of marine compounds. We incubate for about 5 hours to allow for new CCL2 production. Then we take that media and measure how much CCL2 was produced through an ELISA assay. And again, we measured viability by measuring the DNA in the cells. And that has led us to find the compound discodermide. So discodermide is a compound from the sponge Discodermia dissoluta. It has known cytotoxic and antifungal activities, but inhibition of CCL2 secretion by pancreatic cancer cells is a total novel activity for this compound. The IC50, or the dose required to see 50% inhibition for CCL2 of discodermide, is 10 micromolar. The IC50 for CCL2 inhibition of curcumin is 45 micromolar. So again, the marine compound is far more active than the compound that is known to have anti-inflammatory activities. So inhibitors of CCL2 may prevent the mast cells from migrating to the site of the pancreatic where the pancreatic tumor is growing. We also have an assay for finding inhibitors of degranulation. I told you that the mast cells release many soluble factors in addition to the growth factors and angiogenic factors. They release histamine, and they release this enzyme called beta-hexosaminidase. We can induce degranulation by adding the chemical calcimicin, and we hope that in the presence of Harbor Branch compounds, we will see a lot less degranulation. We can take the beta-hexosaminidase that was produced, and we mix that with a different compound, and that produces a light change that we can follow through our plate reader. And that allowed us to find a new activity for the compound topsentin. So topsentin is a compound that we've been very excited about. It has a cytotoxic activity against tumor cells, and it has antiviral activities. We knew that it was a potent anti-inflammatory compound, both in vivo and in vitro, but we didn't know that it acted upon mast cells. And that makes it very exciting because a mast cell influx is not only important for pancreatic cancer, but it has been seen with many other kinds of cancer. So this has potential to be very effective against many different cancers. And the IC50 for degranulation for inhibition of the granulation for topsentin is 11 micromolar. So far we have found 2 pure compounds that inhibit CCL2 secretion by pancreatic cancer cells. We have found 5 pure compounds that inhibit mast cell degranulation, and 27 fractions corresponding to 12 different organisms that require further purification. So we know that mast cells play an important role in pancreatic cancer initiation and progression. We had a small NIH grant that allowed us to do this work, so we only did a discrete screening of about 500 compounds. Our library contains about 5,000, so we want to do more screening because there might be compounds that are even more active than the ones that we have found so far. But the good news is that we have found compounds that inhibit CCL2 secretion by pancreatic cancer cells. We also have found compounds that inhibit mast cell degranulation, and these compounds have the potential to prevent pancreatic cancer from forming, and they also have the potential to treat pancreatic cancer that's already there. And we're hoping to submit a larger grant to screen the compounds that we already find and to find new ones at sometime this year. So now I'm going to tell you about marine natural compounds that affect cell proliferation and survival. And the first compound that I'm going to tell you about is LEIODERMATOLIDE. And some of you might have already heard about this compound because we're very excited about this compound. We try to keep you abreast of the research that we do with it. But this is a potent anti-mitotic agent that was isolated from Lyodermation sponge. It's about 10-fold more active than Taxol, and I told you Taxol is the drug that gets used the most against cancer. I also chose activity not only against pancreatic cancer cells, but against triple-negative breast cancer and against some colon cancers. So one of the ways that we figure out that it is antimitotic is by doing imaging. So these are pancreatic cancer cells that were labeled with an antibody for tubulin, which is the red. And tubulin is very important. It gives the cell its shape. And it also is very important for cell division. You need functional tubulin for that to happen. These cells were also labeled with an antibody for phosphorylated histone H3, which can only be seen in cells undergoing cell division. So this is our control. You can see that there's a few cells that are undergoing cell division, but when you treat with LEIODERMATOLIDE, you cause arrest in cell division, and so you get to see many more cells that are undergoing cell division because you basically stop that cell division from occurring. So we know LEIODERMATOLIDE causes mitotic arrest. This is the boring way which we used to look at apoptosis before we had our imager, but we're looking at the same thing. Cleavage of molecules called caspases is a hallmark for apoptosis, and you can see that LEIODERMATOLIDE cleaves a lot of the caspase 3 and 7 after 24 hours of treatment. And similarly, we look at DNA fragmentation as a hallmark of apoptosis, and you can see that LEIODERMATOLIDE indeed induces this DNA fragmentation in pancreatic cancer cells. So we know that LEIODERMATOLIDE induces apoptosis in pancreatic cancer cells. Most compounds that cause cell cycle arrest do so by acting upon tubulin. And tubulin is constantly coming together and pulling apart. When it comes together, we call that tubulin polymerization. When it pulls apart, we call that depolymerization. Most compounds that are antimitotic affect tubulin one way. So here you have Taxol, and Taxol causes tubulin polymerization, so you can see how much higher that is. We have vincristine and nocodazole, which cause depolymerization, and you can see how they are down in the graph because they didn't polymerize. But if you have tubulin alone, if you have tubulin treated with our vehicle control, or tubulin treated with LEIODERMATOLIDE going from 1 micromolar to 100 micromolar, we had no effects. So we know that LEIODERMATOLIDE does not cause tubulin polymerization or depolymerization. So these are our mechanism of action studies. We know that LEIODERMATOLIDE is a potent antimitotic compound. It has no effect on polymerization or depolymerization of tubulin. We know it affects tubulin, microtubule dynamics, but we don't know exactly where. We haven't been successful at determining exact molecular target if it's binding something, but everything hints at a possible novel mechanism of action. So the bad news is we still do not completely understand its mechanism of action, even though we have tried a lot of things. The good news is that everything that we have tried tells us that this compound is unlike any other compound that causes cell cycle arrest. So that makes it quite unique. And so that is very exciting because it might have the potential to work on those cancers where Taxol doesn't work. And the other good news is that we have a colleague at UCF that has a mouse model of pancreatic cancer, and they put LEIODERMATOLIDE through this mouse model. And at the end of the day, at the end of their study, they measured tumor weight, and you can see that LEIODERMATOLIDE significantly decreased tumor weight when you compare it to control and when you compare it to animals that were treated with gemcitabine. And gemcitabine is the drug that is used to treat pancreatic cancer right now in the clinic. So these are good news. This tells us that LEIODERMATOLIDE not only works on cells, but it works in vivo. So this puts it just one step closer to the clinic. So we're very excited about these results. So LEIODERMATOLIDE so far, we know that it's a potent antimitotic agent that was isolated from the LEIODERMATION sponge. It's about 10-fold more active than Taxol. It has potent cytotoxic activity against pancreatic cancer cells, against triple-negative breast cancer cells, and against colon cancer cells. It causes cell cycle arrest by a very unique mode of action. It affects tubulin dynamics, and it has in vivo and in vitro anti-tumor effects. Now I'm going to talk to you about my baby. I have ugly babies. So this is the molecule MANZAMINE that I've been working with since I came to Harbor Branch, and I am super excited about what this compound can do. So I hope that you will be too after I show you. MANZAMINE has been isolated from cestospongia sponge and haliclona sponge, and we actually believe that it's a microbe inside those sponges that makes that compound. So some of the reasons we got excited about this compound is that MANZAMINE abrogates cell dissociation. And you can see here cancer cells, and they're not touching each other. They like to live on their own, and this is very abnormal. Cells like to touch each other and get their signals out of each other. Look at your skin and look at how close they are. So you can see that the cells alone, they're not touching much, they're not spread out, they kind of look ready to migrate, which is a bad sign. It's a characteristic of a metastatic cell. If you treat these cells with GUO126, which is an inhibitor of PMEC, which is part of the RAS pathway that I showed you earlier, you kind of restore the association of the cells. You can see that now they're spread out, they're touching each other. But you get similar effects if you treat with 2.5 micromolar of manzamine A, and it's much more apparent with 5 micromolar and 10 micromolar of manzamine A, which is about 1/5 of the dose of the other inhibitor. I keep on telling you about apoptosis and our cells being resistant to it. So normally when you have a normal cell, they have what we call dead receptors. And when you bind that receptor with its ligand, in this case, the TNF receptor as apoptosis-inducing ligand or TRAIL, you will induce apoptosis. If you have a cancer cell, though, you can bind the dead receptor with TRAIL, and you still get survival. So what we were hoping is, can we put or pre-treat with a Harbor Branch compound and then bind with TRAIL and see if we can restore the ability to undergo apoptosis? So this is a very busy slide, but what I want you to focus is on this side where the apoptosis is occurring. So in this quadrant, you want to look at the right. The top has the late apoptotic cells, and the bottom has the early apoptotic cells. And so you can see that when I treat with manzamine A, I get about 35% apoptosis for these pancreatic cancer cells. When I treat with TRAIL and methanol, again, I get about 35% apoptosis. But if I treat with a combination of 10 micromolar manzamine A and TRAIL, I get about 85% apoptosis. So manzamine A is not killing the cells itself, but is restoring the ability of the cells to undergo apoptosis. This is very exciting. So next, because we had seen the abrogation of cell dissociation, we wanted to see if it had other anti-metastatic effects. And so we know that cells that are metastatic are able to migrate to other organs. And so we can mimic that in the lab by creating this transwell experiment where we put an insert in our cell culture plate. We can coat that insert with a collagen matrix, and we create what we call a chemotactic gradient by just putting more FBS. We plate our cells on the top, and then we watch them if they make it to the bottom of the plate. So we kind of follow the cells when they're here. And you can see that the cells that are untreated or just receiving methanol are very able to migrate to a membrane alone. They have a little bit more difficulty. We had collagen in there, but they still made it. If we treated with GUO126 or with manzamine A, we took away the ability to migrate. That translates to anti-metastatic properties. So that made us very, very excited. So we knew manzamine A had all these great abilities, but we didn't know how it worked. So we had to start doing mechanism of action studies. And so Dr. Amy Wright at that point had a grant where she was collaborating with Novartis, and they put manzamine A through the HIP-HOP assay. And so HIP stands for haploinsufficiency profiling. You have G strains, which have a single copy of a gene knocked out. Every gene has 2 copies, so we just took down 1. And the strains that are most sensitive to the compound that they're being exposed will become depleted over time. And this kind of, it's illustrated over here. You can see the greens have gone away. And so through this assay, you can identify the compound's target. So if the compound binds something, we will identify it through the HIP. We can also do homozygous profiling, where now we knock both copies of genes that are not essential. And the strains more sensitive to a drug, again, will become depleted, just like this. And this will tell us which genes are required for growth in the presence of the compound. And this is very informative for those compounds that lack a direct binding target. And so this is complicated, but we try to simplify it a little bit. So this is manzamine A, and this is the HIP profiling, and this is the HOP profiling. And under here, we have prodigiosin. And prodigiosin is a compound that is very well studied, and we know it's a known BTPase inhibitor. And you can see that the profiles almost match. So we have no hits on the HIP profiling, so we knew that manzamine A had no direct binding target, but the HOP cluster with known uncouplers of bacular ATPases, and that gave us a hint that that was manzamine A's mechanism of action. So what does that mean? Well, if a cell has uncoupled its bacular ATPases, one of the possible results would be that you will accumulate acidic autolysosomes. And so my postdoc followed this, and he indeed observed that you have that. So here's our vehicle control, and the more green they are, the more brilliant, the more acidic they are. And so here you have cells treated with manzamine A. This is our close-up at 40x. And you can see that there's an accumulation of acidic aerolysosomes after treatment with manzamine A. And this is very similar to baphyllomycin A1 treated cells, that this is a known uncoupler of vascular ATPases. So surprisingly, the combination of both didn't cause even further fluorescence, so we don't know exactly what is going on there. It will require a little bit more studies. But this confirmed that indeed manzamine A uncoupled vascular ATPases. And this led George to think, okay, does that have an effect on autophagy? And autophagy is a pathway that only gets activated under stress, specifically if you're under starvation. So if a cell is undergoing starvation, what it does is it pulls out any organelle that is not using and puts it in a vesicle. Then that gets broken down with an aerolysosome to produce amino acids and fatty acids, and that allows for new protein synthesis and energy production, and the cells is able to survive. So think if you were trapped in a cabin during a blizzard, you will burn anything you need to keep warm. So this is what the cell does. But what cancer does, very smart, it grabs the neighbor healthy cell and breaks it down through this process to obtain nutrients. But so George wanted to find out what happens when we treat cells to autophagy in this pathway. And so we were able to follow LC32, which is a marker of autophagy, that basically the levels increase as autophagy is occurring, and they resolve as autophagy resolves. And you can see this is a Western blot for LC32, and this is LC32. At 24 hours, we got very high levels of it. By 48 hours, they still keep on increasing. If you see under starvation, we induced autophagy to occur, and we saw the increase in LC32, but by 48 hours, it had already resolved. So this led us to conclude that manzamine A was inhibiting autophagy from occurring. So we know that manzamine A is not very cytotoxic on its own, but it has potential anti-metastatic activities because it prevents migration through a collagen matrix. It also reduces cell dissociation. It restores the ability of pancreatic cancer cells to undergo apoptosis. It uncouples vascular ATPases, increasing lysosomal acidity, and inhibits autophagy. So if you recall that pathway that I showed you, the scary pathway at the beginning, so we know inflammation can activate all these signaling pathways, which do all these activities, lead to tumor initiation and metastases. I put manzamine A here in gold because we know that manzamine A inhibits NFKB. It appears to inhibit RAGE. We know it abrogates apoptosis resistance. We know it inhibits autophagy, and it seems to have anti-metastatic properties. So this compound is attacking pancreatic cancer through 5 different ways. So we are super excited about this compound. We hope that this is a compound that makes it to clinic, and that's why I call it my baby, because I'm super excited about the abilities of this compound. So I'm going to tell you a little bit of future directions that we're planning to go. So when we have a cancer cell, how does it go into a full-grown tumor? There's a lot of events that have to happen, but with pancreatic cancer, we knew we could block angiogenesis and the tumor growth still occur. We could cut certain things, and it still managed to grow. And that led us to understand that pancreatic cancer is able to activate very unusual pathways. One of the pathways that it activates is macropinocytosis. We like our big names. So what happens here is that the cancer cell uses its membrane to trap proteins, to create this cup, to trap proteins that are outside of the cell, bring it inside the cell, then it fuses this vesicle with the lysosome, where it gets broken down into amino acids, and that promotes tumor growth. We also know that pancreatic cancer likes to activate autophagy, where it can isolate organelles into an autophagosome, and then this again fuses with a lysosome. They get broken down, they produce amino acids, and produces tumor growth. And as I said, these organelles are likely to be out of the neighboring healthy cell. But again, we see targets. So we are trying to find compounds that block macropinocytosis, And we're trying to find other compounds in addition to manzamine that might block autophagy. And autophagy is going to be important not just for what I told you, but this was the big headline on The Scientist, which is a publication that we get that came out after the last meeting of the American Association for Cancer Research. And what they had found is that in those cells that responded to chemotherapy, and they were starting to undergo apoptosis, if those cells activated autophagy, they were able to thwart the chemotherapy and survive by activating this pathway. So inhibitors of autophagy are going to be very important in fighting cancer. So for macropinocytosis, we have pancreatic cancer cells that we know can activate this pathway, so we're basically just letting it do it, but we're putting a fluorescent tag for it to ingest. And once in there, we are going to follow it using our high-content imager that can image and quantitate the cells. And this is what that looks like. So these are pancreatic cancer cells that were labeled with blue for their nuclei, with green for their body. And those that have internalized the fluorescent tag look kind of pinkish. So you can see all those that have arrows have internalized the fluorescent tag that we put. So the beauty of a high-content imager is that it can quantify this for me. And so we get something that looks like this. We know that our pancreatic cancer cells by themselves have some basal levels of macropinocytosis, but if I put them under conditions of serum starvation, I can really induce a lot of it. If I treat them with manzamine A, it brings that down. So this is a novel activity for manzamine A. I need to confirm it, but it's very exciting. AIPA is a known inhibitor of macropinocytosis. It didn't work that great on this assay. I'm still trying to optimize this assay, but we're very close to having an assay that we can screen for inhibitors of macropinocytosis. For autophagy, we're following a very similar approach to what George did. We are looking at LC32. And so this is what our staining looks. Again, cells are labeled in blue with their nuclei, green for their body, and you can see the red LC32 staining on their membranes. This is a non-specific antibody, and I don't like this differential, so I'm still trying to optimize this staining, which is hopefully going to happen sometime soon. So our conclusions is that we're setting up assays to identify inhibitors of autophagy and macropinocytosis. A discrete screening of about 500 fractions will occur this year. We hope that compounds that inhibit these pathways will cut the few lines of pancreatic cancer, so they will prevent the tumors from growing. And we hope to acquire sufficient data to submit an NIH grant. So to recap, I know I've given you a lot of information, but if you remember these last two slides, you will be okay. So I gave you the scary pathway of progression of pancreatic cancer. So this is inflammation. We know it releases a lot of factors. It activates transcription factors. It activates pancreatic stellate cells, and it attracts cells of the immune system. It activates many cellular events that lead to tumor initiation and metastasis. And hopefully, I have convinced you that we have many different marine natural compounds that are able to inhibit these different events earlier on. And what we hope that will happen is that these compounds can then inhibit all these cellular events to lead to tumor initiation and metastasis. And I just want to acknowledge everybody that worked to get this. George was my former postdoc. He did the work for the mechanism of action of manzamine A, and he also set up the degranulation assay. Tara, Dee, and Pat are staff scientists at the MBBR. They did the bulk of the screening for all the assays that I showed you and also assist me in the mechanism of action studies. Mike, Gervais, Lexi, and Kelly are former summer interns whose work has contributed to these results. And we have our collaborators at Novartis, Dominic Hefner and Tiffany Jag. They ran the hip-hop assay for manzamine A, and Dr. Cheryl Baker at UCF ran our animal studies for LEIODERMATOLIDE. I also want to acknowledge all our funding sources. We are self-funded, so we have NIH grants that support our work. I also got a Florida Department of Health grant that allows us to do a lot of the inflammation work. We are part of the Cooperative Institute, the Cooperative Institute that runs from NOAA, that Shirley Pomponi runs here at Harvard Branch, and that has allowed us to follow some of our mechanism of action studies, and we hope that it's going to help us find collect new samples. The Bernard A. Egan Foundation, the Rose and Gregory Cancer Foundation, and the Lloyd Biddle Popcorn Club have made generous donations to my research. And it's thanks to these donations that I'm able to do those experiments that just take my research one step above. The images that I showed you about apoptosis were submitted as part of my publication to support the data that I had, but they also allow us to follow new directions. So anytime that we find something new about pancreatic cancer, it's thanks to those donations that I'm able to set up assays and try to find those inhibitors. I also want to thank everybody that is part of the MBBR. We're truly a multidisciplinary and collaborative group, so everybody is involved in the effort and the research that I show you. And with that, I thank you, and I'll take any questions.